United States, co-chaired by myself, Sami Khan from University of Otago, New Zealand, and we have a new co-chair, Cleo Stratman from the Netherlands Institute of Ecology. So it's a wide range of people in the GSA, um, and we have an advisor, Dr. Hilary Swan. She's an executive director of Archbold Biological Station. Um, Students in GLEON almost forms one third of the the all GLEON membership, so which is almost 33%, 33%, and one third. GLEON Student Association was founded in 2007 by 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 GLEON students, and I believe that some of those pioneer students are present today. Um, it was formed as a model to train the next generation of scientists within a GLEON, with the main goal that the GSA will be working to inform, train, and mentor students, and also to develop targeted workshops in the GLEON meetings. So mostly focusing on data analysis, modeling, and team sciences. Um, and it's also to create an opportunity for the students to network. The strategy of the GSA is to maintain and improve communication plan, which was primarily through GLEON blogs, but now we are very active on the social media, Facebook and Twitter, and we do organize and trainings for the students. What is GSA has a very like a very active role in the GLEON all hands meeting. One is that we organize this GSA workshop, which is commonly known as Monday workshops, and it's mostly for students and also for the postdoc students, postdoc early career scientists. And we also work with the CCC to form network partnership program called NPP, and we do have a leadership role in the working groups and also in organizing postdoc sessions. NPP, it's it's a very interesting partnership that we encourages like GLEON and GSA encourages new to GLEON partners by pairing the GLEON by pairing the GLEON senior partner with the new one and give them an opportunity to share at least one meal so that they can informally discuss and also to visit the poster sessions together and also to help the new to GLEON in identifying their common interest and also to link the new to GLEON members within to the GLEON senior members and also to the working groups. I won't talk a lot about working groups because you will see many presentations later today about them. We do poster is it's on the second day of the GLEON all hands meeting that the students and also the early career scientists they present their results in a poster and GSA is organizing that. Another activity we have in GLEON is GLEON Fellowship Program, and the very recent fellowship activity we had was a Bayesian statistics workshop, which we organized post G19 at Carey Institute in New York, and it was a group of 13 young scientists from the GLEON students and also postdoc, and they are working on the forecast of gleo blooms and lake canopy, and I was part of that fellowship program, and it sounds like they are now working as a working group, like a full functional working group, and they're gonna come up in the next G20, and they are looking forward to at least publish one paper out of this work. Um, as I said, that we are very active on the social media. So if you are on Twitter, so please don't forget to hashtag GLEON 19.5, and there is also a Facebook page. You can see the link for that. We write GSA blogs to outreach our GLEON community who are not actually active or unable to attend our meetings. So at least that they can also get updates. So in this GLEON blogs, the GSA actually writes GLEON blogs once in a month, or one blog every second month. Our recent blog was about Baz Ebling as a new GLEON co-chair. So if you haven't seen this blog, please visit the GLEON blog page. It's a very inspiring story, at least for me. Um, so as I said, that I will focus mostly on these next step for the GLEON, and I really need from the other GSA leaders as well, that we need your feedback, because we are looking forward to the GSA future vision. Um, so we just want, we see ourselves to assess within the future funding strategies, and also to continue to adopt student needs. Like we just don't want to be like repeating trainings for the students, but we want to adopt that if there are other student needs, so that we can go further and further. Um, and we also want to enhance the member numbers, because we see that this year there were almost 50% less students who applied for the G20, and we just don't know the reason, but that was also one of our concern to think about that. And we also got one feedback from the CCC that the GSA leadership is only 3 people: chair, co-chair, and co-chair elect. And like CCC, we should have ad hoc subcommittees, like for example, a blog subcommittee, or maybe like a GSA workshop subcommittee, something like that. So this is just these are just like the ideas that we just received from the CCC in the last Skype meeting. And there is a new thing that one of our chair will be so we will just announce a GSA co-chair elect position after the G20. And so we have ideas for the GSA G20 workshop. These are just the ideas, but we are still working on it. So there will be QC high frequency data. This will be a training we are just looking forward to, and either a science communication, how to model, how to set up and use models, icebreaker activity, outside activity for students to interact. And this time we also think that we should send at least a survey to the students, that what kind of workshop they need, so that we can organize those workshops for them, just to make our time and our resources very useful for the students. Thank you very much, and I think I'm on time, so I just welcome your questions and also your ideas about our GSA future goals. Thank you very much.
Thank you, Sami. That was perfectly clear to me. Are there any questions for Sami, in particular from maybe students or that are listening in as we speak? Of course, you can always reach out to the GSA with any questions at any time, so it doesn't have to be now. I hear no questions. You can unmute your microphone and you can speak if you want to. If there's nothing there and we're still perfectly on time, then I suggest we move on to the first presentation of today, which is okay. Good luck.
Can you see my screen already?
Could you please increase your microphone? It's a bit low volume.
I try to.
Or just speak clearly into your microphone.
Yeah. Is it better now? Or if I move closer to the computer?
Yeah, it's a bit better. We can hear you. We can up the volumes on our machine.
Okay. Yeah, sorry for that. I'm on a system here that. Okay, I start, not to lose time. So yeah, I really tried hard not to put model into the title. That's maybe why it became a bit long. But while preparing the talk, I actually realized I'm passionate about modeling. And so I will talk more about numerical models and less about tools. But in the spirit of GLEON, please be open, bear with me. And I promise that these will be the only equations that you see in the entire talk. So modeling is a very collaborative process, and it should be very collaborative. And all these people have their fair share in what I'm going to show. And I would like to especially put emphasis on Louise Bruce, who did a lot of the work that I will show later. So from census to prediction, we can see everywhere an increasing availability of data. And we can see that in different research fields and also in our everyday life. And as we could already see yesterday, and we will see today and tomorrow, GLEON is very active and very productive in collecting data and in interpreting data as well. Now, in many of these studies, we see patterns that are rather unexpected. And these studies raise new questions about our process understanding. And that's where numerical models can be a really helpful tool to understand aquatic ecosystems. So with models, we can test our knowledge, we can test and create hypotheses, and we can extrapolate in space and time. So that means if you have data gaps, a model might help you to bridge those gaps. Or if you just are able to sample at one location or 2-3 locations in the lake, then a model can help you to get a bit more spatial information. And what's the really strength of models is that they deliver prognosis, or they are able to deliver prognosis. And that is increasingly used by lake and reservoir managers. And it has already saved a lot of money and resources. So if you talk to a few reservoir managers, they have good examples where models were really useful. So what I would like to emphasize here, that we should see models more as a complementary tool to lab experiments, to field studies, and to statistical analysis. So they don't solve everything in your life. Now, I would like to talk about a model that is really deeply rooted within GLEON, and it's called GLM, or the General Lake Model. Don't mix it up with the generalized linear model from statistics. So GLM here is a numerical model, and it's a hydrodynamic lake model. And for the talk, I've compiled a short history of GLM. I won't go through every detail of it. If you want, you can have a look later because the talk is recorded. So around 2010, there were a few people, and I think David Hamilton was among them, and Matt Hipsey, and they felt that there's a need for an open, one-dimensional hydrodynamic lake model. And that's how the idea of GLM was born. So GLM is supposed to simulate water temperature and stratification. And 2 years later, the first version was tested, and the legend tells that in a pub in the evening, the highly excited modelers, they actually came up with another idea. So they said, why shouldn't we take the model and apply it to many different lakes and see how it goes? So that's how the GLM MSCP project was born. And I think the same year GLEON got on board. Now, quite a few years later, finally we have the publication of the GLM MSCP, and we can celebrate a really successful project. And I would like to talk a bit about it. So MSCP, it's a multi-lake comparison project. And the whole project was really collaborative. So it was between two different networks. The other network is AMON, which is a network of aquatic ecosystem modelers. And it was a collaboration between data custodians and modelers. And the idea was that we use data that we already have and that we apply the model to as many lakes as we can get, so where we can get the data from. And we ended up with 32 lakes ranging across all different climate zones and in morphometry. So you can see here that the largest lake, for example, is Lake Namco in Tibet. And then compared to that, the smallest lake is Blelam Tarn in the English Lake District. So it's really a wide range of different lakes and reservoirs. Now, the idea was to interpret water temperature and stratification and see how well the model performs, and also to conduct a parameter sensitivity analysis. So changing a bit the settings of the model and seeing what happens with the model results. So for the results, I won't go too much into detail because it's a rather technical paper. It's really good for people who start using GLM or other one-dimensional hydrodynamics models, because you can compare your lake to the lakes that the model was used to, and you can see a bit about the parameters that you have to set. What the study was really good for is so-called model benchmarking. So by applying the model to many different lakes and reservoirs, we can get an idea where is GLM performing well and where are the applications maybe a bit problematic. And this is really useful, for example, for managers to see if they can rely into a model. Now let me show you some results. Each of these blue abbreviations is one lake. And then on the y-axis, we have the root mean squared error. Now that's a metric to tell you a bit how well your model performs. So the smaller the value, the better the model. And if the model would be perfect, all the points would be 0 for y. And on the x-axis for the left to left figures, we have the extinction coefficient. So that tells you how much of the light is attenuated in the water column. So it's a measure for turbidity or for water quality. And we can see some trends in the model results. So we see that with an increasing extinction coefficient, the model is less good in predicting the full temperature profile, and the pattern is even more clear for hypolynean temperatures. Now that is relatively easy to explain because GLM is just a hydrodynamic model, so it ignores seasonal patterns in phytoplankton growth, or if there's a turbidity event, it won't grasp that. So this might be improved by coupling the model with some water quality model. And what's also interesting to see is on the right hand, you see the results plotted over depths. So it looks like GLM is performing better for deeper lakes and less good for deeper lakes and less good for shallow lakes. And that could relate to the model structure. And actually there have been some model developments going on now, and soon there's a new model released that is performing also, or that is also applicable to very shallow lakes. So these studies help us to see where do our models need improvements, and they help us to learn about the model, but also about the lakes. And it was a really exciting project. And what was really nice about the project as well, there was a lot of parallel development going on. So GLM is now open source. You can find it on GitHub. And there's an active user forum. And here I would like to say kudos to Luke Winslow, who's really answering a lot of the questions arising. And what's also nice to see in the forum is that people who were new to GLM a few years ago, they are now helping and replying to people who are now beginning to use it. GLM, there were also some R packages developed to facilitate the use of the model. And we also wrote an introductory chapter how to use the model. Now, all this makes the model easier to use. And I just want to alert you a bit, be aware of best practices, because the easier it is to use a model or a tool, the easier it is to misuse a model or a tool. And we can see that there's a lot of discussions, for example, in statistics as well. So there's a lot of information out there on best practices, And what's really good in this respect is the project that Kaylin Carey was mainly initializing. So she and her colleagues, they developed some teaching modules for students, and one of them is about GLM and how to use models. So from early on, it educates students what they can do with models and what they should take care of. Now I would like to show you that we have to be really careful about model results with two examples. So one is focusing a bit on parameterization and uncertainty. Koji was so nice to give me some of his simulation results that he did. And he had a look at a water quality model, and he took the parameters for the water quality model. This could be something like phytoplankton growth rate. And he tried to assess the uncertainty in these parameters and look what happens with the model results if now I increase that uncertainty. Because we have to give a fixed value to the model, but actually phytoplankton growth rates or nutrient uptake, they can vary, and they vary between species. So if you increase the uncertainty in your parameters, obviously the result in your model and the uncertainty in your model will increase as well. So that's something you have to be aware of as well when applying modeling studies. And that's something we should work on to improve our water quality models. And the second example is on structural uncertainty. It's from my PhD thesis. So I looked at, I compared basically two different processes for nutrient limitation of phytoplankton growth. And the only, that was the only difference in the two different model versions. And you can see a large result in the phosphorus distribution in Lake Constance, which is a deep lake over depth and over time. And now if I would give this to the lake manager, that could result in quite different ideas of what to do with that lake. So also here we have to be really careful. What's the model structure? What do the equations imply that I'm using? And I think GLEON could be a really good network to refine our water quality simulations. And we have a new project, Operation Equation, that's the first step in that direction. But we will talk about that in the working group a bit more tomorrow. So coming back to the history of GLM, we can see that there were a lot of versions released over the years. And that's actually pretty nice because it tells us that the model has improved quite a lot and further developed quite a lot. But that raises also a question. Who develops and tests and maintains the models and tools? And in the case of GLM, this is basically the aquatic ecodynamics group around Matt Hipsey who are doing that. And I wonder a bit if that's a system that we can sustain and that we want to sustain, and how can we do that? Or do we want to change it, and can we change it? So let me show you that a colleague of mine, she put it very nicely that there are different types of modelers. So there are the designers, you can see them a bit as the architects of the model. And then there are the programmers who sit down and put all the ideas into machine code. And then there are the users who apply the model and run scenarios and so on. So for model development, we need the designers and the programmers, but there are not so many people who can do that. And also, how can we give them credit? For example, a programmer might be a computer scientist who doesn't need to have papers, but then a designer might be somebody who can get credit from papers. So that's something we can think about. And we should also think about studies like the one by Stora et al. That show that actually a collaboration between all of them and between also other scientists, like people working in the lab, in the field, that the collaboration will result in better code. And what as GLEON, I think we should also do is we should think in workflows. So from the very initiation of a project, think about what software do I need to read the sensor data and to transfer them to do the quality control, to do the analysis. How can we link the different steps, and let people who are good in technology, let them help you, talk to them, and don't think you have to rewrite everything. There's a lot out there. And maybe we could think about a system to acknowledge software development, code development. There's a lot of discussion going on authorship, and there's a system called credit, which I find really useful. Maybe we could think about something similar in the modeling and in the software domain. At least try to cite papers that are linked to the software, and I think GLEON is doing really, really well in this respect. And I emphasize this paper by Jordan Reed et al. That looks at it from the GLEON perspective in a really interesting way. So what's going on at the moment, and where are we moving? A current project that's now upcoming is the GLEON EasyMap, and it's really exciting. It's a project where we want to apply different models to run climate change scenarios. And what's nice about it and exciting is that if we are good and if we are fast, we can make it into the next IPCC report. And the project is really open to everybody. It's open for data custodians, it's open for modelers, it's open for people who are willing to handle large data sets. And the good thing is that the next info meeting is actually tomorrow. So just jump on, the contacts are given here. And we will talk about it in the working group tomorrow as well. And also as a little advertisement, we are planning to run a small AIM&J workshop. So a workshop for aquatic ecosystem modelers that are students, PhD candidates, postdocs. And we sort of doing that right before GLEON, so you don't have to pay extra travel costs. And we try to keep it really low cost. And yeah, we can talk with the student association as well. Maybe we can collaborate there. So with that, I would like to end with a famous quote. All models are wrong, but some are useful. And thank you for your attention.
Thank you, Marieke, for a nice talk. Are there any questions for Marieke? Anywhere? Very quiet. Then maybe I can ask a question, Marieke. You mentioned a few times the need to integrate observations, experiments, and models. And thinking back, you know, I used models mainly in the years that I was working as a lake manager or consultant, because you don't have labs, so models are your main tool. So my view would be, and I would ask you if you agree with that, that, for instance, if you look at lake managers, the sort of the really new insights, they often come from observation and experiments. Think, for instance, why do we control phosphorus in lakes? Initially, we didn't know. So there was the experiment by Schindler, there was the observation by Vollenweider pointing at phosphorus as the culprit for eutrophication. So would I be correct in saying that models maybe generate less really new information, but are very useful in making the information that is there available in an applied sense, maybe? Or would you completely disagree with that view?
I would agree to a certain extent. I would say it depends on which kind of model you're looking at. So the minimalistic models that are used in theoretical ecology, I think they are more exploratory, and yeah, people get ideas from that. And maybe the models we use in management, the lake ecosystem models, they are more used in a way to describe processes and then to test different scenarios. I wanted to say something else. I lost my train of thoughts. Gone. Damn it.
It's gone. Anything else?
Yeah. I wanted to say, I think the models help us to look at our systems from a different perspective. And that can also generate ideas about experiments and generate ideas about new field campaigns. Yeah. So it's sometimes good. And what I see working with colleagues with, for example, real phytoplankton ecologists, talking to him, we have different ways of thinking, different ways of looking at stuff, and working together is actually, I think, really productive.
Can I add something, Bas?
Yeah, sure.
So I don't Well, I think I agree with what you said, but I think there's a lot more to it than that. And um, I think the models can do things sometimes that experiments and field observations can't do. And I want to give an example. So when I was working for the New York City water supply, we were interested in looking at the impacts of climate change on the drinking water reservoirs. And there's two impacts. There's the impact of climate on the watersheds and the loadings to the reservoirs, and there's also the impacts on the thermal structure and the mixing and all the chemical and biological dynamics in the reservoirs. And by using coupled models, we could do something that you couldn't do in reality, is that we could hold the watershed loads to present conditions and allow the climate change to only impact the reservoir or vice versa. We could look at only the watershed effects while holding the effects of climate change on mixing process and thermal structures constant. And that's really valuable. And the only way you can do those kinds of things that I know of is by doing modeling. So I think the models allow you to look at things in a very different way and perform sort of numerical experiments that just aren't possible in reality. So I think there's a very, very valuable source there as well.
Good, thanks, Don. I was mainly trying to provoke some discussion, but I would agree that one hand washes the other. So we need all three of them to really make progress. I think, thanks Marika again. I think it's time to move on, looking at the time in the schedule.
I have a short question. Are these talks available? It would be nice to have them for references.
They will be. They're being videoed and they will be available on the GLEON website.
Okay, good. That's it.
Announced. Okay, Jens.
How do I share my screen? Okay, I see it. Sorry.
Yeah. I guess it takes a minute from what I understand.
Yes, it's been known to take a few seconds to respond. Yeah, it's coming. Yep, you're there.
You can all see it?
Yep, thanks, Greg.
Okay. All right, well, first of all, thank you all for being here. I've become interested in changes in deep water oxygen depletion in lakes, and I'd like to tell you a little bit about this and how I became curious about the phenomenon. And I want to do this through asking questions. This is going to be more about the development of a GLEON project than it is about giving a lot of answers and scientific data. I want to start, though, by reinforcing some of the things that Sandy said this morning in his introduction, and Bas yesterday, and that is the value of GLEON to graduate students. This is a group of my past and present graduate students who have been very actively involved in GLEON. And to a great extent, graduate students run GLEON, which is a real pleasure to see. These students here have been GLEON fellows, they've been GSA chairs, they've been steering committee members, they've led working groups, they've published manuscripts. Most importantly, they've grown tremendously and become, I think, really outstanding scientists from this. So thank you, GLEON, and all of you involved. So I'm going to talk a little bit about transparency regulation of oxygen in lakes, or the so-called TROLL project, where we hope to use high-frequency sensors. In fact, we're starting to do this already to look at oxygen depletion deep in lakes. And this is a list of the current participants, but there are new people joining all the time. It is a green project, so please join us if you're interested. We'll have a working group meeting after this presentation. So it's all about the questions. Curiosity, why do algae grow? We know light, water, and nutrients. We don't pay much attention to water in lakes because there's plenty of water. If somebody asked you what is important in controlling algae, you'd probably say nutrients. In fact, you'd probably say nutrients, and wouldn't pay a whole lot of attention really to light. And in fact, if you think about it, I think limnology kind of has a holy grail in terms of lake management for algae blooms. And this is the log chlorophyll versus log total phosphorus relationship, which is very compelling. Increase phosphorus and you increase algal blooms. You decrease phosphorus and you decrease chlorophyll. Classic study by Tommy Edmondson back in 1970. actually in the 60s, published in 1970. Whole lake experiment by David Schindler. Add carbon and nitrogen to one side of a lake, carbon, nitrogen, and phosphorus to the other, you get algal blooms. Nitrogen, of course, is important too. Jim Elser did these studies of hundreds of lakes and marine environments, as well as terrestrial, and you add a little bit of nitrogen, you get an increase in nitrogen. Add a little phosphorus, you get an increase in phosphorus. Add both, and you get a bigger increase. So bottom line is nutrients seem to be very important in lakes. No surprise. So how have nutrients and chlorophyll changed in lakes in recent decades? Well, one of the most comprehensive studies to date of almost 3,000 lakes by Samantha Oliver and others analyzed changes in nutrients and chlorophyll in the northeastern United States and found an unexpected stasis in a changing world in terms of lake nutrient and chlorophyll trends. Main message was basically no overwhelming degradation or improvement. So as important as nutrients are, there's a pattern of stasis. I think an interesting GLEON project would be extend this pattern from northeastern United States to a global scale. But if nutrients and chlorophyll are static, what is changing in lakes? And one of the things, anybody who knows me knows I deal with browning in lakes and have a strong interest in this. And we've seen dissolved organic matter or dissolved organic carbon increases in many lakes around the world. Fennoscandia, 67% increase, and DOM in almost 500 lakes that are looked at. The two graphs here show an approximate doubling or even greater than doubling of dissolved organic matter in some lakes on this side of the pond. We've seen, a number of us anyway, have seen this paper by Monteith et al. In Nature showing an increase in dissolved organic matter in Northern Europe. Northeastern North America, the same place where Oliver et al. Did their study and found no surprising change in nutrients, there in the Midwest anyway. Increase in browning in many of the lakes there. This little red X here is a location of a couple of the study sites that I'll be talking more about in just a minute. In terms of why lakes are browning, I'm not going to go into the details here. There are a lot of reasons for browning, but two of the primary ones are increase in precipitation, which we have seen in our lakes in the northeastern United States. In fact, it's one of the areas of the world where we're seeing the most rapid increases in extreme storm events, which are very important to DOM loading in lakes. But in addition, we're recovering from an acid rainstorm, acid precipitation storm. Here again, that X is the location of the study lakes I'll be talking about in a minute. You can see in 1990, high levels of hydrogen ion deposition. Moved to 2014, thank you, Clean Air Act amendments, drastic reductions in hydrogen ion deposition. And together, these increases in precipitation in the northeastern United States, combined with this decrease in acid deposition, have really contributed to browning. So what? How does browning alter lake ecosystems? What are some of the effects? Let's take a look at two of our study lakes, Giles and Lackawanna. This is a blue water lake and a brown water lake. Semi-brown water lake, only about 5 milligrams per liter. Some people wouldn't call it that brown, but it's certainly browner than Lake Giles. Located in northeastern United States, both less than 50 hectares. If we look at some of the changes that are going on, you can see, first of all, the browning in Lake Giles, the blue lake, more than a doubling of dissolved organic carbon concentrations. With that, there's been a tremendous recovery in pH. Remember the acid rainstorm from which we're recovering. Same sort of patterns in Lake Lackawanna, but not as pronounced, the browner lake. Changes in water transparency. Maybe light's important here. 10% PAR depth, photosynthetically active radiation, used to be on the order of 14 or 15 meters. It's now about 6 meters. Lackawanna, same kind of pattern, but not as pronounced. Chlorophyll, not really changed too much. Giles, at the moment, the pattern is significant, but due largely to this single point at the end. The point is there's been a lot of scatter, not a pronounced change in chlorophyll in either of these two study lakes. One of the most interesting changes has been the depletion of oxygen in the deep waters. This is the example from Lake Giles. And what you can see is there has been a decrease of on the order of 0.2 milligrams per liter per year in the deep waters of Lake Giles, highly oligotrophic lake that is now going anoxic by the end of the summer. And so this is really what has largely driven my own curiosity about what's going on deep in lakes, and the recent development of advanced sensors that automate the collection of dissolved oxygen and temperature in the deep waters of lakes now enables us to look at the short-term dynamics of oxygen in lakes, and that's kind of what this GLEON project is all about. We have a nice foundation in Steve Jane's, or the GLEON project is led by Steve Jane on long-term oxygen. This is some of his preliminary data from over 200 lakes that shows the decrease in deep water oxygen in somewhere on the order of 58% of the lakes. Now, these aren't all significant, but the pattern seems to be declining oxygen across a number of lakes globally, not just in our few local lakes. So how widespread is this dissolved oxygen depletion? In catchments with no land use change in particular. One thing I didn't mention, but I should have, is our two study lakes are very well protected watersheds, and there's no land use change. So it's not a matter of people throwing more fertilizer on the farms or leaky septic systems, that type of a thing. We're getting this oxygen depletion for some other reason. Excuse me. So we're particularly interested in the phenology of light availability, and does light availability play a role? And how does incident PAR even vary? A lot of people still use the old dinner plate, the Secchi disc, to look at water transparency, but hopefully we're getting beyond that at this point. So how does PAR, photosynthetically active radiation, vary seasonally or interannually on the same day? These are data over a 20-year period looking at incident. So this is above the water surface solar radiation coming into the lake. And as you might expect, you have much higher levels in the middle of the summer than you do in the winter. And there's a lot of variability on a day-to-day basis. If you look at the seasonal variation, it's on the order of 10-fold from summer to winter. So broad variations in incident solar radiation. Even if you look on a single day of the year, and among years, you see a large variation of greater than 3-fold in incident solar radiation, dependent on cloud cover primarily. And this is our view, limnologist view, most limnologist view, usually through a Secchi disc, of what's going on with light in lakes. And what you can see is we're looking at a small snapshot of the peak of the incident solar radiation time period here. There are other things that control solar radiation too. What about ice cover? And this really gets us into the heart of the Troll project. Ice cover period in inland lakes and rivers has really changed. Nice paper by Barbara Benson, who was very active in early years of GLEON, by the way, found that duration of ice cover is shortened by about 30 days. And moreover, the trends are about 2.5 times greater in the past 30 years than they were in the preceding years. So changes in ice cover obviously will alter light availability in lakes as well. Let's put these pieces together. This is some modeling done by my PhD student, Rachel Pilla. And this is the black line here, the big smiley face that goes across the whole screen, is what you would get in terms of the depth of average summer 1% surface irradiance. So this is how deep 1% of the summer surface irradiance goes all different times of the year. So you have less light in the incident light in the winter, and you also have not only less incident light, but you have the potential for snow cover. Clear ice, white ice, snowy ice, and snow-covered ice can block almost all of this solar radiation. And the depths that are shown here are the 1% average summer photosynthetically active radiation. So this is the approximate compensation depth. If we're deeper than that, there's net dissolved oxygen consumption. If we're shallower than that depth, there's net dissolved oxygen production. So the main thing here, the main point here is we are missing out by looking at the average summer 1% PAR depth. In fact, most of us even use a Secchi disk. At least that's what we see in many of the published papers still. We're looking at a very, very small point in this larger smile. And what about ice cover and the changes in ice cover? How does this change the incident light cover? If you have snow-covered ice, again, you can eliminate pretty much all of the light getting down into a lake. And this might be important not only to algal productivity, but to regulating decomposition and the balance between dissolved oxygen production and consumption. So here's the conceptual model we have for the TROLL project. Past phenology, we're looking from winter to spring to summer to autumn. We have period of winter ice cover. We have a mixing period. We have summer stratification. In response to climate change, we're seeing a shorter period of winter ice cover, shown in red here, in response scenario number 1. We might see an extended stratification during the summer as a result of that, with no change in the mixing periods. This would lower the dissolved oxygen, increase the depletion. Another response scenario, though, is we might get an increase in the mixing periods, which would increase the ventilation, nicely shown by Raoul Couture and others. It may not be a matter of increased depletion of oxygen. It might be a higher dissolved oxygen concentration. So the questions we're asking in this TROLL research project are, what role do changes in the phenology of ice cover and these alternating periods of stratification and mixing have specifically on light availability, and what role do they play in determining the phenology of the deep water dissolved oxygen? And we can look at this now that we have some really nice advanced sensors. We're also interested in what effect do weather events have on the availability of dissolved oxygen. This might be heat waves, cold snaps, droughts, floods, high versus low wind periods, hurricanes, typhoons, and those sorts of things. Our approach is going to be to use these advanced sensors, not only for dissolved oxygen and temperature, but we hope turbidity, chlorophyll, CDOM sensors. Many of us have these sensors and they're in place now. We're collecting data. Some of us have done it for a year or two, some even more. Pilot studies are underway. The project is wide open, it's green, and you're welcome to join us. That's pretty much all I have to say. Thank you for listening.
Thank you, Craig. Any questions? Anywhere in the world?
Well, we could come with a comment here. We're very interested in this project since we are doing experiments in our large lake lab with Browning. We have all these sensors that have been running for several years. I saw you had Hans-Peter Grossart in, so he has maybe told you something about that. But that's in the middle of, we are doing large-scale mesocause experiments on, in the middle of the Lake GLEON, which we also have all the sensors in. And there is also, of course, a work from IGB here on this other lake, which is divided in 4 pieces, which is differently brown. So anyway, very interesting. We will certainly like to join.
Great.
Yeah.
Thanks, Jens. I had a quick question, Craig, on the stasis. So you say chlorophyll tends to sort of stay constant, but there is a clear trend, an increase in brownification. So intuitively, I would think, well, if light is getting less, there's less opportunity for algae to produce biomass, so that might reduce chlorophyll. At the same time, the size of the antenna would go up in the cell, so that would increase the amount of chlorophyll per cell. Do you think that there's some reason for the stasis in chlorophyll, that there are two potentially contrasting effects there?
Yeah, very, very good point. Chlorophyll is not really the best measure of either biomass. I mean, it's the most useful one and most widely available, but there certainly are issues with using chlorophyll. I think the main thing is that we see a lot of noise in the chlorophyll, but we see some really pronounced changes in the dissolved organic carbon, in the water transparency, and the pH, and there are some really substantial changes that are occurring, and I think that there's pretty good evidence for light controlling these. In fact, if you look at a couple of papers that just came out, one by Rachel Pilla in Geophysical Journal of Geophysical Research, Biogeochemical Sciences, and another one in Inland Waters by Leslie Knoll, you'll see these changes in transparency have really influenced the thermal stratification and the oxygen in these study lakes here. Whether that's true around the world, I don't really know. I'm sure that in some places, in fact, if you look carefully at Samantha Oliver's paper, you'll see there certainly are lakes where chlorophyll is changing and nutrients are changing. It's not as if there are no change to any lakes. And this is the reason for the Troll project, is to look into this.
Okay. Thank you very much, Craig, for your presentation. I think we should move